Hi everyone, I'm Jen, one of the prac tutors for Advanced Immunology. I'm also a second year PhD student in Professor David Hume's macrophage biology group, which means I spend a lot of time looking at these guys, tissue resident macrophages. Now, in order to do this, I have to use immunohistochemistry, which is more commonly referred to as IHC. And today, I wanted to have a chat with you about direct versus indirect IHC. So, as you probably know, immunohistochemistry exploits the principles of antibodies binding specific antigens in biological tissues. And it also makes use of chemical reactions in which color changes take place. It's most commonly used when we want to know about the spatial location of the antigen or the cells that we're interested in terms of where they are within the tissue sample that we're looking at. In the prac today, you're going to be using direct detection immunohistochemistry. In particular, you're going to use an F480 antibody. So the antibody recognizes F480, which is a very commonly used marker for macrophages and monocytes. It's already conjugated to a molecule called biotin, and this helps you see where that antibody has actually bound within the tissue after a few more chemical processes. But I want you to imagine that you're all ready to do your experiment and you go to the antibody box only to find that someone's used all of the anti-F480 biotin conjugated antibody. Not a very good start to your day, especially because you really need to get this experiment done. Knowing that, you go off to the lab antibody inventory to see if you have any other suitable antibodies. You find that, in fact, you do have another F480 targeting antibody that will target mouse F480. And note that this antibody was raised in rat. However, it's not conjugated to a fluorophore or biotin. That means you're going to have to perform indirect detection. Now, what this means is, as well as your primary antibody, you're going to need a secondary antibody. And it's the secondary antibody in this case that has a molecule conjugated to it that's eventually going to enable you to see where the primary antibody bound in the tissue. For example purposes, I'm going to stick with biotin because that's what you're doing in prac. But what should our antigen binding site of this secondary antibody actually be targeting? Well, remember I said the primary antibody was raised in rat, so it's a rat IgG targeted against mouse F480 antigen. That means that for the secondary antibody, its antigen binding site should be raised against rat IgG. Most secondaries are raised in animals such as goats, sheep, donkey, maybe even rabbit. But let's say it was raised in goat. What you would actually be looking for is an antibody that's called goat anti-rat IgG biotin conjugated antibody. It's a bit of a mouthful, but it's really important that you understand what each part of that name is referring to. So what's going on in the tissue when we're doing this staining process? Well, you'll have the macrophages with the F480 on the cell surface, and we incubate with the primary antibody. So this will bind really tightly to the F480, and then we can add the secondary antibody. So more than one secondary can bind to the primary. And then we have these biotin molecules, but the biotin isn't visible to us. We need to add a molecule called streptavidin, which binds biotin. Then we add a biotin horseradish peroxidase conjugate. The horseradish peroxidase is really important because it's what causes a color change when we add DAB. So DAB in the presence of HRP will become a brown precipitate. So overall, this process is letting us see where that primary antibody bound. Now, of course, more than one of the conjugates can bind to the streptavidin. More than one streptavidin can bind to these secondary antibodies because there'll be multiple biotin molecules. And then more than one secondary antibody can bind to the primary antibody. So we've got multiple levels here where we're enhancing the signal of that primary antibody. That's really important when you're looking for antigens that may not be expressed particularly highly. So now that we've gone through all of these different steps, we can see our beautiful macrophages. Here's an example of some that I stained in a kidney section. But in order to get the most out of our tissue and the spatial information, it's best that we stain the tissue with a counterstain. Often this will be something such as hematoxylin, or as you use today, toluidine blue. And these bind the nucleic acids in the nucleus and give us a much better picture of the cytoarchitecture of the tissue and where our antigen of interest is within that. So just to recap, today you're doing direct IHC because the antibody that you add that targets the antigen of interest is already conjugated to biotin. An antibody example for this would be a rat anti-mouse F480 biotin conjugated antibody. But in indirect IHC, we need to use a primary antibody such as rat anti-mouse F480, and then we need our secondary antibody such as a goat anti-rat IgG conjugated to biotin, and this will let us see where the primary bound within the tissue. Hopefully you realize that because both the molecules, both the antibodies have biotin, we still do the streptavidin and horseradish peroxidase and DAB step for both of these. And I hope that this makes direct and indirect IHC a bit clearer for you. Of course, if you have any questions, do ask your tutors and have a chat with them. Okay, bye.